Glycolysis is the metabolic pathway that converts glucose C6H12O6 into pyruvate, CH3COCOO H+. The free energy released in this process is used to form the high-energy compounds ATP and NADH. Glycolysis is a determined sequence of 10 enzyme-catalyzed reactions. The intermediates provide entry points to glycolyses. For example, most monosaccharides, such as fructose and galactose, can be converted to one of these intermediates. The intermediates may also be directly useful. For example, the intermediate dihydroxyacetone phosphate is a source of the glycerol that combines with fatty acids to form fat. Glycolysis occurs, with variations, in nearly all organisms, both aerobic and anaerobic. The wide occurrence of glycolysis indicates that it is one of the most ancient known metabolic pathways. Indeed, the reactions that constitute glycolysis and its parallel pathway, the pentose phosphate pathway, occur metal-catalyzed under conditions of the Archean Ocean, also in the absence of enzymes. Glycolysis could thus have originated from chemical constraints of the prebiotic world. Glycolysis occurs in most organisms in the cytosol of the cell. The most common type of glycolysis is the Indena-Uromahufa-Uropanase, which was discovered by Gustav Impton, Otto Meyerhoff, and Jakub Karel-Panase. Glycolysis also refers to other pathways, such as the Entner-Urodowdorov pathway and various heterofermentative and homofermentative pathways. However, the discussion here will be limited to the Indena-Uromahufa-Uropanase pathway. The entire glycolysis pathway can be separated into two phases, the preparatory phase in which ATP is consumed and is hence also known as the investment phase, the payoff phase in which ATP is produced. Overview: the overall reaction of glycolysis is, the use of symbols in this equation makes it appear not balanced with respect to oxygen atoms, hydrogen atoms, and charges. Atom balance is maintained by the two phosphate groups. Each exists in the form of a hydrogen phosphate anion, dissociating to contribute 2 hours plus overall. Each liberates an oxygen atom when it binds to an ADP molecule, contributing 2O overall. Charges are balanced by the difference between ADP and ATP. In the cellular environment, all three hydroxyl groups of ADP dissociate into O and H+, giving ADP3A, and this ion tends to exist in an ionic bond with Mg2+, giving ADP-Mg. ATP behaves identically except that it has 4 hydroxyl groups, giving ATP-Mg2A. When these differences along with the true charges on the two phosphate groups are considered together, the net charges of a 4 on each side are balanced. For simple fermentations, the metabolism of 1 molecule of glucose to 2 molecules of pyruvate has a net yield of 2 molecules of ATP. Most cells will then carry out further reactions to repay the used NAD+ and produce a final product of ethanol or lactic acid. Many bacteria use inorganic compounds as hydrogen acceptors to regenerate the NAD+. Cells performing aerobic respiration synthesize much more ATP, but not as part of glycolyses. These further aerobic reactions use pyruvate and NADH+H+ from glycolyses. Eukaryotic aerobic respiration produces approximately 34 additional molecules of ATP for each glucose molecule, however most of these are produced by a vastly different mechanism to the substrate-level phosphorylation in glycolyses. The lower energy production, per glucose, of anaerobic respiration relative to aerobic respiration, results in greater flux through the pathway under hypoxic conditions, unless alternative sources of anaerobically oxidizable substrates, such as fatty acids, are found. Elucidation of the pathway. In 1860, Louis Pasteur discovered that microorganisms are responsible for fermentation. In 1897, Eduard Buchner found that extracts of certain cells can cause fermentation. In 1905, Arthur Hardin and William Young along with Nick Shepard determined that a heat-sensitive high molecular weight subcellular fraction and a heat-insensitive low molecular weight cytoplasm fraction are required together for fermentation to proceed. The details of the pathway were eventually determined by 1940, with a major input from Otto Meyerhoff and some years later by Louis Leroy. The biggest difficulties in determining the intricacies of the pathway were due to the very short lifetime and low steady-state concentrations of the intermediates of the fast glycolytic reactions. Sequence of reactions, preparatory phase. The first 5 steps are regarded as the preparatory phase, since they consume energy to convert the glucose into 2,3-carbon sugar phosphates. Payoff phase. The second half of glycolysis is known as the payoff phase, characterized by a net gain of the energy-rich molecules ATP and NADH. Since glucose leads to 2 triose sugars in the preparatory phase, each reaction in the payoff phase occurs twice per glucose molecule. This yields 2 NADH molecules and 4 ATP molecules, leading to a net gain of 2 NADH molecules and 2 ATP molecules from the glycolytic pathway per glucose. Regulation. Glycolysis is regulated by slowing down or speeding up certain steps in the glycolysis pathway. This is accomplished by inhibiting or activating the enzymes that are involved. The steps that are regulated may be determined by calculating the change in free energy, Ig, for each step. If a step's products and reactants are in equilibrium, then the step is assumed not to be regulated. Since the change in free energy is zero for a system at equilibrium, any step with a free energy change near zero is not being regulated. If a step is being regulated, then that step's enzyme is not converting reactants into products as fast as it could, resulting in a buildup of reactants, which would be converted to products if the enzyme were operating faster. Since the reaction is thermodynamically favorable, the change in free energy for the step will be negative. A step with a large negative change in free energy is assumed to be regulated. Free energy changes. The change in free energy, Ig, for each step in the glycolysis pathway can be calculated using Ig equals Ig degree plus RTLNQ, where Q is the reaction quotient. This requires knowing the concentrations of the metabolites. All of these values are available for erythrocytes, with the exception of the concentrations of NAD+ and NADH. The ratio of NAD+ to NADH in the cytoplasm is approximately 1000, which makes the oxidation of glyceraldehyde 3-phosphate more favorable. Using the measured concentrations of each step, and the standard free energy changes, the actual free energy change can be calculated. From measuring the physiological concentrations of metabolites in an erythrocyte, it seems that about 7 of the steps in glycolysis are in equilibrium for that cell type. 3 of the steps the ones with large negative free energy changes are not in equilibrium and are referred to as irreversible. Such steps are often subject to regulation. Step 5 in the figure is shown behind the other steps, because that step is a side reaction that can decrease or increase the concentration of the intermediate glyceraldehyde 3-phosphate. That compound is converted to dihydroxyacetone phosphate by the enzyme triose phosphate isomerase, which is a catalytically perfect enzyme. Its rate is so fast that the reaction can be assumed to be in equilibrium. The fact that Ig is not zero indicates that the actual concentrations in the erythrocyte are not accurately known. Biochemical logic. The existence of more than one point of regulation indicates that intermediates between those points enter and leave the glycolysis pathway by other processes. For example, in the first regulated step, hexokinase converts glucose into glucose 6-phosphate. Instead of continuing through the glycolysis pathway, this intermediate can be converted into glucose storage molecules, such as glycogen or starch. The reverse reaction, breaking down, for example, glycogen, produces mainly glucose 6-phosphate. Very little free glucose is formed in the reaction. The glucose 6-phosphate so produced can enter glycolysis after the first control point. In the second regulated step, phosphofructokinase converts fructose 6-phosphate into fructose 1,6-bisphosphate, which then is converted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The dihydroxyacetone phosphate can be removed from glycolysis by conversion into glycerol 3-phosphate, which can be used to form triglycerides. On the converse, triglycerides can be broken down into fatty acids and glycerol. The latter, in turn, can be converted into dihydroxyacetone phosphate, which can enter glycolysis after the second control point. Regulation. The three regulated enzymes are hexokinase, phosphofructokinase, and pyruvate kinase. The flux through the glycolytic pathway is adjusted in response to conditions both inside and outside the cell. The rate in liver is regulated to meet major cellular needs, the production of ATP, the provision of building blocks for biosynthetic reactions, and to lower blood glucose, one of the major functions of the liver. When blood sugar falls, glycolysis is halted in the liver to allow the reverse process, gluconeogenesis. In glycolysis, the reactions catalyzed by hexokinase, phosphofructokinase, and pyruvate kinase are effectively irreversible in most organisms. In metabolic pathways, such enzymes are potential sites of control, and all three enzymes serve this purpose in glycolyses. Hexokinase. In animals, regulation of blood glucose levels by the pancreas in conjunction with the liver is a vital part of homeostasis. In liver cells, extra G6P may be converted to G1P for conversion to glycogen, or it is alternatively converted by glycolysis to acetyl-CoA and then citrate. Excess citrate is exported to the cytosol, where ATP citrate lyase will regenerate acetyl-CoA and OAA. The acetyl-CoA is then used for fatty acid synthesis and cholesterol synthesis, two important ways of utilizing excess glucose when its concentration is high in blood. Liver contains both hexokinase and glucokinase. Both catalyze the phosphorylation of glucose to G6P, but the latter is not inhibited by G6P. Thus, glucokinase allows glucose to be converted into glycogen, fatty acids, and cholesterol even as G6P accumulates in hepatocytes. This is important when blood glucose levels are high. During hypoglycemia, the glycogen can be converted back to G6P and then converted to glucose by the liver-specific enzyme glucose 6-phosphatase and released into the blood without taking up the low concentration of glucose it releases. This reverse reaction is an important role of liver cells to maintain blood sugars levels during fasting. This is critical for brain function, since the brain utilizes glucose as an energy source under most conditions. Phosphofructokinase. Phosphofructokinase is an important control point in the glycolytic pathway, since it is one of the irreversible steps and has key allosterate effectors, AMP and fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate is a very potent activator of phosphofructokinase, which is synthesized when F6P is phosphorylated by a second phosphofructokinase. In liver, when blood sugar is low and glucagon elevates CAMP, PFK2 is phosphorylated by protein kinase A. The phosphorylation inactivates PFK2, and another domain on this protein becomes active as fructose bisphosphatase 2, which converts F2,6BP back to F6P. Both glucagon and epinephrine cause high levels of CAMP in the liver. The result of lower levels of liver fructose 2,6-bisphosphate is a decrease in activity of phosphofructokinase and an increase in activity of fructose 1,6-bisphosphatase, so that gluconeogenesis is favored. This is consistent with the role of the liver in such situations, since the response of the liver to these hormones is to release glucose to the blood. ATP competes with AMP for the allosterate effector site on the PFK enzyme. ATP concentrations in cells are much higher than those of AMP, typically 100-fold higher, but the concentration of ATP does not change more than about 10% under physiological conditions, whereas a 10% drop in ATP results in a 6-fold increase in AMP. Thus, the relevance of ATP as an allosterate effector is questionable. An increase in AMP is a consequence of a decrease in energy charge in the cell. Citrate inhibits phosphofructokinase when tested in vitro by enhancing the inhibitory effect of ATP. However, it is doubtful that this is a meaningful effect in vivo, because citrate in the cytosol is utilized mainly for conversion to acetyl-CoA for fatty acid and cholesterol synthesis. Pyruvate kinase. This enzyme catalyzes the last step of glycolysis, in which pyruvate and ATP are formed. Regulation of this enzyme is discussed in the main topic, pyruvate kinase. Post-glycolysis processes. The overall process of glycolysis is, glucose plus 2 Namibian dollars plus 2 ADP plus 2 pyruvate plus 2 NADH plus 2 hours plus 2 ATP plus 2 hours 2O. If glycolysis were to continue indefinitely, all of the NAD plus would be used up, and glycolysis would stop. To allow glycolysis to continue, organisms must be able to oxidize NADH back to NAD+. How this is performed depends on which external electron acceptor is available. Anoxic regeneration of NAD+, one method of doing this is to simply have the pyruvate do the oxidation. In this process, pyruvate is converted to lactate in a process called lactic acid fermentation. Pyruvate plus NADH plus H plus a lactate plus NAD+. This process occurs in the bacteria involved in making yogurt. This process also occurs in animals under hypoxic conditions, found, for example, in overworked muscles that are starved of oxygen, or in infarcted heart muscle cells. In many tissues, this is a cellular last resort for energy. Most animal tissue cannot tolerate anaerobic conditions for an extended period of time. Some organisms, such as yeast, convert NADH back to NAD+ in a process called ethanol fermentation. In this process, the pyruvate is converted first to acetaldehyde and carbon dioxide, then to ethanol. Lactic acid fermentation and ethanol fermentation can occur in the absence of oxygen. This anaerobic fermentation allows many single-cell organisms to use glycolysis as their only energy source. Anoxic regeneration of NADH is only an effective means of energy production during short, intense exercise, providing energy for a period ranging from 10 seconds to 2 minutes and is dominant from about 10–30 seconds during a maximal effort. It replenishes very quickly over this period and produces 2 ATP molecules per glucose molecule, or about 5% of glucose's energy potential. The speed at which ATP is produced is about 100 times that of oxidative phosphorylation. The pH in the cytoplasm quickly drops when hydrogen ions accumulate in the muscle, eventually inhibiting enzymes involved in glycolysis. The burning sensation in muscles during hard exercise can be attributed to the production of hydrogen ions during a shift to lactic acid fermentation as oxygen is converted to carbon dioxide by aerobic respiration faster than the body can replenish it. These hydrogen ions form a part of lactic acid along with lactate. The body falls back on this less efficient but faster method of producing ATP under low oxygen conditions. This is thought to have been the primary means of energy production in earlier organisms before oxygen was at high concentration in the atmosphere and thus would represent a more ancient form of energy production in cells. The liver later gets rid of this excess lactate by transforming it back into an important glycolytic intermediate called pyruvate. See Cori cycle. Fermentation of pyruvate to lactate is sometimes also called anaerobic glycolysis. However, glycolysis ends with the production of pyruvate regardless in the presence or absence of oxygen. Anaerobic respiration. In the above two examples of fermentation, NADH is oxidized by transferring 2 electrons to pyruvate. However, anaerobic bacteria use a wide variety of compounds as the terminal electron acceptors in cellular respiration, nitrogenous compounds, such as nitrates and nitrites. Sulfur compounds, such as sulfates, sulfites, sulfur dioxide, and elemental sulfur. Carbon dioxide. Iron compounds. Manganese compounds. Cobalt compounds. And uranium compounds. Aerobic respiration. In aerobic organisms, a complex mechanism has been developed to use the oxygen and air as the final electron acceptor. First, pyruvate is converted to acetyl-CoA and CO2 within the mitochondria in a process called pyruvate decarboxylation. Second, the acetyl-CoA enters the citric acid cycle, also known as Krebs cycle, where it is fully oxidized to carbon dioxide and water, producing yet more NADH. Third, the NADH is oxidized to NAD+ by the electron transport chain, using oxygen as the final electron acceptor. This process creates a hydrogen ion gradient across the inner membrane of the mitochondria. Fourth, the proton gradient is used to produce about 2.5 ATP for every NADH oxidized in a process called oxidative phosphorylation. Intermediates for other pathways. This article concentrates on the catabolic role of glycolysis with regard to converting potential chemical energy to usable chemical energy during the oxidation of glucose to pyruvate. Many of the metabolites in the glycolytic pathway are also used by anabolic pathways, and, as a consequence, flux through the pathway is critical to maintain a supply of carbon skeletons for biosyntheses. In addition, not all carbon entering the pathway leaves as pyruvate and may be extracted at earlier stages to provide carbon compounds for other pathways. These metabolic pathways are all strongly reliant on glycolysis as a source of metabolites, and many more. Gluconeogenesis, lipid metabolism, pentose phosphate pathway, citric acid cycle, which in turn leads to amino acid synthesis, nucleotide synthesis, tetrapyrrole synthesis. From an anabolic metabolism perspective, the NADH has a role to drive synthetic reactions, doing so by directly or indirectly reducing the pool of NADP+ in the cell to NADPH, which is another important reducing agent for biosynthetic pathways in a cell. Glycolysis in disease, genetic diseases. Glycolytic mutations are generally rare due to importance of the metabolic pathway. This means that the majority of occurring mutations result in an inability for the cell to respire, and therefore cause the death of the cell at an early stage. However, some mutations are seen, with one notable example being pyruvate kinase deficiency, leading to chronic hemolytic anemia. Cancer. Malignant rapidly growing tumor cells typically have glycolytic rates that are up to 200 times higher than those of their normal tissues of origin. This phenomenon was first described in 1930 by Otto Warburg and is referred to as the Warburg Effect. The Warburg hypothesis claims that cancer is primarily caused by dysfunctionality in mitochondrial metabolism, rather than because of uncontrolled growth of cells. A number of theories have been advanced to explain the Warburg effect. One such theory suggests that the increased glycolysis is a normal protective process of the body and that malignant change could be primarily caused by energy metabolism. This high glycolysis rate has important medical applications, as high aerobic glycolysis by malignant tumors is utilized clinically to diagnose and monitor treatment responses of cancers by imaging uptake of 2-18F2-deoxyglucose with positron emission tomography. There is ongoing research to affect mitochondrial metabolism and treat cancer by reducing glycolysis and thus starving cancerous cells in various new ways, including a ketogenic diet. Interactive pathway map. Click on genes, proteins and metabolites below to link to respective articles. Alternative nomenclature. Some of the metabolites in glycolysis have alternative names and nomenclature. In part, this is because some of them are common to other pathways, such as the Calvin cycle. See also. References. External links. A detailed glycolysis animation provided by IUBMB, the glycolytic enzymes in glycolysis at RCSBPDB, glycolytic cycle with animations at wdv.com, metabolism, cellular respiration and photosynthesis, the virtual library of biochemistry and cell biology at biochemweb.org, notes on glycolysis at rehalbledwin.com, the chemical logic behind glycolysis at UFPPT, XPASY biochemical pathways poster at XPASY, mnemonic medicum mnemonics.com 317 5468.